Hi, my name is Ellie Clifford, and this is my Ecological Integration Synopsis report over 3 different student presentations, which I will discuss in this video. The first presentation I attended was by Abir Chaudhary, who is a graduate student at Texas A&M. The title of his presentation was Urbanization at the Cost of Biodiversity, Examining Urban Growth on Habitats and Key Biodiversity Areas in Indonesia. Since the rate of habitat loss and fragmentation has been increasing in developing countries across the world due to urban land expansion, Chaudhary looked specifically at the effects this had on Indonesia, as it is a developing country with growing biodiversity. For his research question, he asked where recent urban land expansion caused the largest habitat loss across the country, which species were the most effective, and if the protective status of key biodiversity areas made any difference in terms of habitat loss to urban land expansion. Chaudhary executed this question by studying urban growth over a 23-year period from 1992 to 2015, as well as its impact in habitats across Indonesia, specifically around and in the key biodiversity areas. Through this study, Chaudhary determined that during the studied period, urban land increased by 67.5% in the country and also increased by about 8% within its key biodiversity areas. The islands that showed the highest urban expansion rates in Indonesia were Sumatra with a 124% increase and Kalimantan with a 99% increase. Most shockingly, Chaudhary found that the key biodiversity areas in Indonesia that were disrupted by urban land expansion had increased by 30% during this period, with the protected key biodiversity areas suffering more of an impact than the unprotected areas, having an increase of 37% and 29% respectively. Chaudhary also reported that the island that experienced the greatest impact from urban expansion on key biodiversity areas, species habitats, and biodiversity was Java, which was initially the largest urban area in the country. I found this study to be quite interesting but not entirely shocking. I am not surprised that urban land expansion is constantly causing decreasing biodiversity since, as noted earlier, this expansion leads to habitat loss and fragmentation, although it is saddening to learn how urban growth continues to weaken biodiversity in other countries as well. Since I grew up in Houston, a large metropolitan city, I am always seeing areas that provide habitats for wildlife being destroyed in order to expand urban areas. There were constant removals of woodland areas to make room for housing districts or highways. Despite its disheartening effects on biodiversity, sadly, the results from this study are often seen in all of our daily lives. The next presentation I attended was by Ziyu Wang, a graduate student and research assistant in the Department of Biology at Texas A&M. The title of this presentation was Abiotic and Biotic Factors that Shape Cassiopeia Organism-Environment Interaction in Response to Global Change. This presentation described how Wang aimed to understand the organismal reactions to environmental change, specifically how the Cassiopeia is able to thrive in a human-disrupted environment. Furthermore, Cassiopeia has seen to be an invasive species in northern Florida, and through their analysis are trying to realize how abiotic and biotic factors promote this range expansion. In order to examine this, the first thing Wang did was use iNaturalist data to model the degree of range expansions, phenotypic variations, and habitat dynamics. Secondly, Wang examined the role of light and intensity that triggers metamorphosis, specifically on strobulation, where the jellyfish will asexually reproduce by splitting into smaller segments. The findings from the analysis indicate that there was indeed an expansion northward through Florida, and that habitat variability may influence phenotypic diversity. Through some controlled lab experiments on polyps, where different groups were exposed to different amounts of sunlight, Wang concluded that strobulation increased with light exposure. Ultimately, the Cassiopeia is uniquely impressive in its ability to adapt to diverse environmental conditions. I found this study to be more surprising than the last. I personally do not have that much prior knowledge on Cassiopeia or other species of jellyfish, but after a quick Google search, it is apparent that jellyfish are in a steady increase due to the warming of the oceans. Regardless of that information, I am still surprised by this study, as I would not have expected jellyfish, Cassiopeia in particular, to be one of the species that was able to thrive despite global changes. The next presentation I attended was Alana Fulkerson, who is a graduate student at Texas A&M. The title of this presentation was Environmental DNA Analysis for Detection of Poeciliopsis in Northern Sonario, Mexico. This presentation described how several species of Poeciliopsis are found in Sonario State, Mexico, threatened by habitat loss and invasive species. In particular, the Poeciliopsis jackschultzii, or more commonly known as the Rob Conception top minnow, are endangered according to the IUCN Red List, and it is unknown if they are even still in existence. This conveys the issue of the inability to use external morphology to differentiate P. jackschultzii from other similar species with the same genus. The Poeciliopsis occidentals, or more commonly known as Gila top minnows, and Poeciliopsis monica, more commonly known as headwater top minnows. In order to resolve this issue, Fulkerson tested the use of a metabarcoding environmental DNA-based method to test teleosts as a non-invasive alternative for biodiversity and ecosystem monitoring. This method called for collecting and filtering water samples in 2022 and 2023 from a site that was historically known to have been home to the Poeciliopsis being researched. DNA was then extracted from the samples and were put through polymerase chain reaction with a primer set that targeted the specific taxa of interest. This resulted in finding about 238 million reads associated with 2,925 unique features, although none of the unique features matched to P. jackschultzii, signifying the absence of or population below detectability of this species. I found this presentation to be quite insightful and informative. I think that alternative ways to monitor biodiversity and ecosystem health in a non-invasive manner is very helpful for future uses. If this method were to continue to advance and become more precise, it would have a very significant impact on how we are able to monitor ecosystems. In my Ecology 205 lab, we conducted an experiment to determine the overall health of different ecosystems in College Station and Bryan, Texas. However, in order to execute this lab, our methods were very invasive to the surrounding species, having to wade through the streams, physically picking up different organisms, or having to add things into the stream, all disrupting the natural state of the streams. I believe that this study provides a much more suitable way to monitor ecosystems. Thank you.